So in the last class, we have seen second week development. So this is the third week development. So as we have already set the format that changes in the embryoblast and changes in the trophoblast. So changes in the embryoblast are formation of primitive streak, gastrulation, that is formation of the three germ layers. The third germ layer here is the intraembryonic mesoderm. Then the formation of notochord, allantois, so neural plate, neural tube, neural crust, and then the somites, and then the intraembryonic coelom. So in the trophoblast, there is formation of secondary and tertiary villi. You can see at the end of the second week, you can see here clearly there is a bilaminar germ disc, there is an amniotic cavity, and there is a definitive yolk sac. And here is the connecting stalk. This is extraembryonic mesoderm. So these are the three main events in the third week. The gastrulation, it's a formation of the trilaminar germ disc. Primitive streak. Primitive streak is appearing on the 15th day. It's a thickened linear band of epiblast at caudal end of embryo on dorsal aspect of embryonic disc. So this is the indication of beginning of a gastrulation. So what is primitive node, otherwise called Hensen's node or primitive knot? It's an elevated cranial end of primitive streak that surrounds a depression called primitive pit or blastophore. So this Kolar sickle or Robert sickle is the thickening of extraembryonic tissue at caudal edge of the germ disc, which induces primitive streak formation in the epiblast. So nodal gene initiates and maintains primitive streak formation. You can see here, so amniotic cavity has this amniotic cavity is exposed here, showing the dorsal aspect of the embryonic disc. You can see on the cranial end there is oropharyngeal membrane, on the caudal end there is primitive streak, and this will be the primitive node. You can see here this is the hypoblast and this is the epiblast. Electron microscopic view, you can see here the primitive node and primitive pit, and this is the primitive streak. You can see the detaching cells from the primitive streak. So this is the epiblast and this is the hypoblast. So primitive streak regresses by 26th day after contributing for lower part of ventral abdominal wall. Then early regression causes ventral body wall defects, whereas incomplete regression, that is persistence of primitive streak after 26th day, causes sacrococcygeal teratoma. You can see here this is the sacrococcygeal teratoma. Gastrulation. So the germ disc and primitive streak elongate cranio-caudally. The primitive groove develops. This is the central depressed area of primitive streak. Epiblast cells migrate towards primitive streak and starts invaginating. These cells displace hypoblast and form definitive endoderm. So that means from the epiblast, few cells are going to the hypoblast and displacing these hypoblast cells and forms the endoderm. And after that, this migrating cells from the epiblast form the third layer between endoderm and the epiblast called intraembryonic mesoderm by epithelial mesenchymal transformation. And the remaining cells of the epiblast form the ectoderm. So there is a formation of ectoderm from the epiblast, there is formation of intraembryonic mesoderm from the epiblast, and there is also formation of the definitive endoderm from the epiblast. So there is a formation of trilaminar germ disc, which is called as gastrulation. You can see here these are the epiblast cells migrating from the primitive streak, and see few cells are coming to the hypoblast and replacing the endoderm, and here these are migrating as the intraembryonic mesoderm between the epiblast and the hypoblast. So epiblast forms all the three germ layers. The migrating mesodermal cells establish contact with the extraembryonic mesoderm at lateral end of the germ disc. So this mesodermal cells establish contact with the extraembryonic mesoderm also at the lateral end of the germ disc. So intraembryonic mesoderm is divided into three parts. One is the paraxial mesoderm, which is next to the midline, and this forms the somites. Intermediate mesoderm, which is responsible for development of urogenital system in between, and then on the lateral aspect there is lateral plate mesoderm, which splits into somatopluric and splanchnopluric mesoderm with the development of intraembryonic coelom. I'll show you in the next diagram. So mesoderm separates ectoderm from endoderm except at two places: cranially the buccopharyngeal membrane or the oropharyngeal membrane, and caudally the cloacal membrane. You can see here, so this is the developing intraembryonic mesoderm, which is divided into a paraxial mesoderm on either side of the notochord, and you can see this is the intermediate mesoderm and this is the lateral plate mesoderm. You can see here these are the small cavities appearing in the lateral plate mesoderm. So once the cavity is established, this is divided into a somatopluric mesoderm and the splanchnopluric mesoderm. Splanchnopluric mesoderm lines the definitive yolk sac, and somatopluric lines the amniotic cavity. And now you can see here the buccopharyngeal membrane. It's a oval depressed area at cranial end. It appears in precaudal plate. It is bilaminar. So here ectoderm is adherent to endoderm. So breakdowns in fourth week to form stomodium or oral cavity. Cloacal membrane, again small circular bilaminar area at the caudal end of primitive stick. Ectoderm is adherent to endoderm. So divides into urogenital membrane anteriorly and anal membrane posteriorly. So it disintegrates in 7th week to form genital tracts, urethra and anal opening. You can see here, this is the oropharyngeal membrane or the buccopharyngeal membrane, and this is the cloacal membrane. And cranial to this, there will be formation of pericardial bar, which is a mesodermal condensation. So horseshoe-shaped mesodermal cell condensations in cranial to buccopharyngeal membrane is the pericardial bar. So then what happens to these germ layers? Ectoderm gives rise to epidermis, CNS, venous and retina. Endoderm gives rise to epithelial lining of the respiratory tract, GIT and glands opening into them, liver, pancreas and gallbladder. So mesoderm forms the musculature, bone, cartilage, reproductive and excretory system and cardiovascular system. This is an overview. So how these germ layers are giving rise to general structures in the body. Notochord, it's a midline structure developing during 3rd week, develops from epiblast cells on 17 to 18 day, extends from primitive streak to precaudal plate. Cells of primitive knot proliferate and form a depression, primitive pit or blastopore. Proliferation of cells from primitive node forms solid notochordal process. Extension of the primitive pit into notochordal process forms the notochordal canal. The cells of the notochordal canal fuse with endoderm. So cells of the notochordal canal disappear from forming communication between amniotic cavity and yolk sac. This is called neuroenteric canal because there is a rapid differentiation occurring in the epiblast. So there it needs more nutrition. So this neuroenteric canal helps the yolk sac to supply nutrition to the rapidly changing epiblast. So neuroenteric canal flattens to form notochordal plate in the roof of the yolk sac. Then notochordal groove is formed from the notochordal plate. Cells of the notochordal groove get separated from the endoderm and later solidified to form definitive notochord. Significance: so it is a characteristic of phylum Chordata and defines various axes, right left axis and dorsoventral axis, and then forms basis for developing bodies of the vertebra and acts as primary inducer. The nucleus pulposus of intervertebral disc and apical ligament of tens are the remnants of notochord in humans. Remaining part disappears. You can see here from the primitive node, the cells are proliferating to form the notochordal process, and you can see the cavity of primitive node, that is the primitive pit, is extending into the notochordal process, forms the notochordal canal. Then you can see this cells which are adherent to the hypoblast or the endoderm, they are disintegrating, creating a communication between the yolk sac and the amniotic cavity, that is the neuroenteric canal. So this is the neuroenteric canal, and you can see again this neuroenteric canal is closed by this flattening of the notochordal process, so which forms the notochordal plate. Then further the notochordal plate again depresses to form the notochordal groove and finally gets detached from the endoderm to form the definitive notochord. You can see here is the communication between the yolk sac and the amniotic cavity through neuroenteric canal, the notochordal plate, and here you can see the definitive notochord. So allantois, you can see here this is the allantois, extension of yolk sac into the connecting stalk. 16-day finger-like outpouching of yolk sac into the connecting stalk connected to cloaca. So this is actually occurring during the formation of the tail fold. Absorbed partly into primitive urinary bladder, remaining part forms urachus, which is seen as the median umbilical ligament. Significance: so in birds and reptiles it is a reservoir of urine. Allantoic blood vessels form umbilical blood vessels. So developmental animals are urachal, cyst, sinus and fistula. You can see here. So this is the allantois connected to the cloaca. This is the allantois. You can see here this is the urachal fistula. So there is a continuous tract between the urinary bladder and the umbilicus. So urine will extravasate from here, and you can see here closed urachal cyst, and you can see urachal sinus opening at the umbilicus, but it's closed on the other end. So fistula, cyst and sinus. So here you can see the establishment of the craniocaudal and dorsoventral axis. So this is the anterior visceral endoderm, which is defining the cranial end. And here you can see the gosicoid, caudin, noggin and holstein. So this will help in the differentiation of the or dorsalization of the mesoderm in the cranial end. And brachyury T will help in dorsalization of mesoderm on the caudal region. bone morphogenetic protein 4 will help in ventralization of the mesoderm. So you can see overexpression of glycosid, which is defining the dorsalization of the mesoderm in the cranial region. When there is overexpression of glycosid, so it results in conjoint tens. So axis formation, primitive stick formation on dorsal aspect of the bilaminar embedded disc caudally defines craniocaudal axis, medial and lateral and interdorsal axis. The nodal gene, member of the transferrin growth factor beta, continues to express on left side, which further induces expression of transcription factor PTX2, which determines right-left axis. You can see here the nodal being expressed on the left side, and further nodal FT2 and PTX2 determining the left side. On the opposite side, there is no clear-cut identification of specific factors here. So here you can see from the primitive stick the cells migrating from the most cranial part of primitive stick, the cells migrating from the notochord. Next to that they form paraxial mesoderm, next to that they form intermediate mesoderm, next to that they form lateral plate mesoderm, and next to that they form extraembryonic mesoderm. You can see the cyanomelia. So loss of mesoderm in the lumbosacral region resulted in fusion of limbards. So this condition is called cyanomelia. Now you can see here, so at the end of the second week there were primary villi only. Now you can see the extraembryonic mesoderm projecting into the primary villi to make them or to convert them into secondary villi. You can see here, this is the primary villus with syncytium and cytotrophoblast. So with the mesoderm it becomes secondary, and with the blood vessels it becomes tertiary. You can see here, these are the tertiary villi being formed, and you can also see the cytotrophoblast has gone to the outside of the trophoblast. It covers the entire trophoblast and is in direct contact with the desictia basalis. You can see here, so this is the tertiary villi. So this is actually the anchoring villi, and these are the branching villi. This is the intervillar space. You can see the cytotrophoblast has reached to the outside and it has formed a complete shell. And clearly you can see this is the maternal blood vessels which are entering into the intervillar space. Formation of the neural tube. So this is called neurulation. The process of formation of the neural plate groove folds and closure of these folds to form the neural tube. So after gastrulation, ectoderm is divisible into two parts. The midline neuroectoderm which overlies notochord and surrounding surface ectoderm. So thickening of the ectoderm between primitive node and precaudal plate in the midline forms neural plate. So elevation of the edges of the neural plate forms neural folds laterally and central depressed neural groove. You can see here, this is the neural plate, and you can see the elevated lateral margins forming the neural folds and the central depressed area neural groove. And here you can see certain somites are there. Paraxial mesoderm has started forming somites. The neural fold starts fusing in cervical region at the level of the fifth somite and extends craniocaudal to form neural tube. So end of neural tube are called cranial and caudal neuropores. Cranial neuropore closes by 25th day at 20 somite stage. Caudal neuropore closes on 27th day at 25 somite stage. During detachment of the neural tube, cells from margin of the neural fold separate and lies between neural tube and ectoderm called the neural crest cells, which undergoes epithelial to mesenchymal transformation. You can see here, this is the closure of the neural tube. Here you can see this is the anterior neuropore and this is the posterior neuropore. Neural crest is the lateral margin of the neural plate at its junction with the surface ectoderm. Forms dorsal mass and ventral mass. From the dorsal mass, if you see the neuroblast cells of the dorsal mass, they form the dorsal root ganglia or odontoblasts, sensory ganglia of the 5, 7, 19 cranial nerves, parafollicular cells of thyroid, skeletal elements of the pharyngeal arches. And then spongioblasts form the Schwann cells, satellite cells in the ganglion, pluripotent cells form the melanocytes. Ventral mass, also called as sympathochromaffin organ. So sympathoblasts, they form the sympathetic and parasympathetic ganglia. Chromaffin cells of internal medulla, paratic body, surgentaffin cells in respiratory system, and interchromaffin cells in GIT. Other derivatives include the facial bones and vault of skull, dermis of the face and neck, muscles of ciliary body, smooth muscles to blood vessels of face and forebrain, sclera and choroid of the eyeball. You can see here, so the lateral margin of the neural plate or the neural groove, you can see the neural crest. It is getting separated and lies between the neural tube and the surface ectoderm. okay, which is giving rise to cells like sympathetic ganglia into the interchromatic tissue, and you can see the preaortic ganglia, intraganglia. Thank you.